It's a pleasure to present an update on the developments at Cytovia Therapeutics at the annual European Life Sciences CEO Forum. So Cytovia was formed in the summer of 2019 with vision that cell therapy, and particularly natural killer cells, could dramatically change the outcome of cancer treatment. So if we look at the last 10 years, cell therapy has really been a revolution. In the last 10 years, you have 2 Nobel Prizes awarded to relevant technology in 2012, IPSC induced pluripotent stem cell and the ability to differentiate them into adult cells, including immune cells. And then in 2020, gene editing was the Nobel Prize attributed to the inventors of CRISPR. And at the same time, if you look from a clinical standpoint, in 2012 is the first CAR-T clinical trial at UPenn, and it's interesting to note that several of the early patients we had life expectancy of a few months are still alive in 2022. In 2017, the first CAR-T was approved. And when we were formed in 2019, NK cells were still under the radar screen, and it's really the first publication by Kadi Rezvani at MD Anderson in the New England Journal of Medicine in early 2020, as well as the initial clinical data from FATE that changed the outlook for NK cells. And then just last year in 2021, the combination of NK Engager antibodies with NK cells, which is very relevant to the Cytovia strategy. So Cytovia was formed around 2 main platforms, iPSC-derived NK cells with the ability to gene edit the stem cells prior to differentiation and expansion, and FlexMK cell engager antibody, which are multifunctional antibodies targeting both the immune cells, in our case, the NK cell, and the tumor cell. Our goal is not just to have platform technology, but it's to develop therapeutics that address unmet medical needs. Our first program is toward hepatocellular carcinoma or liver cancer, and it's a first-in-class targeting GPC3, both with the engagers and with the cells. Our first INDs are expected to be filed before the end of 2022. We also have a broader pipeline that includes cells and engagers, targeting CD38 for myeloma and EGFR for glioblastoma. All of that has become possible through a combination of, initially multiple alliances, building an open architecture R&D ecosystem. But in the last 6 to 9 months, we've built internal R&D and manufacturing capabilities. We've also strengthened significantly the management team, and in partnership with institutional investors in China, we have built a new company called Cytovia to address the opportunity of Greater China. From our 2 platforms, we can develop 4 types of products: unedited iNK cells, edited iNK cells, CAR iNK cells, with the CAR serving as a GPS to target the tumor, and FlexMK cell engager antibodies. And we can address not only hematological malignancies, but also solid tumors. This is an overview of our pipeline, and you can see that our primary focus is around the GPC3 targets, and the 2 IND that we intend to file in 2022 are for CYT303, which is a GPC3 FlexMK Engager antibody, and CYT100, which is an unedited NK cells, and we intend to evaluate them separately as well as in combination. In 2023, we'll introduce edited iNK cells to address some of the challenges of the tumor microenvironment, as well as the GPC3 CAR iNK. Additionally, as I mentioned earlier, we have a pipeline with CD38 and EGFR products. Our ecosystem is formed of leading academic institutions such as University of California, San Francisco, the National Cancer Institute, in the US, but also INSERM in Europe and the Hebrew University of Jerusalem. We've also collaborated early on with the New York Stem Cell Foundation in New York City. They really helped us with the development of differentiation from iPSCs into NK cells. We also have partnership with Cellectis for their talent gene editing, and with Cytovia Therapeutics for the EGFR CAR NK. Cellectis, Cytovia, and NICEF are actually shareholders of Cytovia. And you can see here in a picture of our team in Natick, Massachusetts, as well as a schematic of what our cell manufacturing facility in Puerto Rico will look like when it opens in the coming months. So why China? China is the second largest market in the pharmaceutical market in the world, and not just in number of patients, but also in dollar or euro value. Liver cancer, which is our lead indication, is 10 times more prevalent in China than it is in the US or in Europe. So there's an opportunity to accelerate the development of our lead program and include China in our global development program. There's also an opportunity, thanks to the platform that we've built in China, to attract other advanced therapeutics for development and commercialization in China. And then CytoLynx may have the opportunity to develop its own products for global commercialization. And very proud of the quality of the investors that have supported us for CytoLynx. We have multiple value inflection points in the next 2 years, and some of them are related to data being available and data being presented at oncology conferences. The first 2 abstracts that we submitted to AACR, the American Association for Cancer Research, have been accepted, and we look forward to presenting them in April. We also expect to have data presented at many of the oncology conferences in 2022, 2023, and beyond. From a regulatory standpoint, at least 2 INDs in 2022 and multiple additional ones in 2023 and 2024, thanks to the platform. The platform is also an opportunity for Cytovia to engage in multiple partnership with other biotech companies as well as big pharmas. So let's focus on the GPC3 hepatocellular carcinoma program. We see the HCC market as a blue ocean opportunity, and unfortunately, the unmet medical need is substantial, and 72% of patients are non-responsive to the best standard of care today, which is Atezo and Beva, and the progression-free survival is only 6.8 months. As I mentioned earlier, there's a large number of patients in Asia, and particularly in China, but substantial also in the US and in Europe. We see the HCC market changing very quickly. First of all, significant growth for the checkpoint inhibitors, moving to first and second line, but also increasingly bispecifics and cell therapy. GPC3 is a very novel, very interesting target for solid tumors. In HCC, it's overexpressed, actually more than 66%. I think some more recent publications are are suggesting as much as 80%, and particularly in more severe patients. But other solid tumors, including squamous lung cancer, also express GPC3. We're not the only one, developing GPC3 cell therapy. That includes Takeda, Adicet and Regeneron, Cytovia, and ourselves, but we're the first one to have iPSC-derived NK cells and CAR NK cells, and also the FlexMK Engagers. Our GPC3, binder was licensed from the National Cancer Institute in the US, the lab of Mitchell Hall. There are multiple high-quality publications of Mitchell Hall, and the fact that it binds to HYP7, which is the proximal side of the receptor, seems to make a significant difference. So we're using the GPC3 binder, first of all, in our FlexMK Engager, which you can see a schematic on the left-hand side. So we're targeting the HTC cells through GPC3, and we're targeting the NK cells through NKp46. We have in vitro functional activities and demonstration that we're binding to both the NK cells and the HTC cells, but we also have 2 in vivo studies, which will be presented at the upcoming meetings in the spring. This is the design of our phase 1/2, and as you can see, we're evaluating both monotherapy and combination, and we expect to have initial data in 2023. So let's move to our platform technologies and manufacturing. We have fully integrated in-house process development and cGMP manufacturing capabilities for our NK cell platform. This was developed in our labs in Natick, Massachusetts, and is being transferred to the GMP facility in Puerto Rico. We've demonstrated both in vitro and in vivo the cytotoxicity of our iNK cells. IPSC Technology, this is a very heavy slide, but I want to say that it's an evolution. You have a first generation of autologous CAR T and evolution to allogeneic. And we believe that iPSC derived NK cells and potentially other types of cells are the best standard today and going forward. And this is because of safety, but also it's available off the shelf. There's unlimited scale up of the manufacturing. There's the ease of editing on the stem cells prior to differentiation. Our collaboration with UCSF and Dr. Equim is around the optimal loci to insert the CAR, and will evolve toward finding new costimulatory domains on the CAR. Our collaboration with Cytovia is using TALEN gene editing, and Cellectis is developing custom talents for Cytovia towards specific targets. This is a very collaborative partnership where Cellectis has been investing significantly, and Cytovia is basically taking their proven technology with T cells into the field of NK cells. Our multi-specific platform, as I mentioned earlier, binds to both the tumor cells and the NK cells through NKp46. The proprietary mutations allow for alignment of the heavy chains and the light chains. We've demonstrated, stability, manufacturability, and because it's tetravalent, very high avidity and improved affinity. And then the Flex linker facilitates the binding to multiple antigens on different cells. We chose NKp46, primarily because it's consistently expressed not just on hematological immunological tumors, but also in infiltrating NK cells, for solid tumors. If you look at the other companies developing NK engagers, we're the only company together with Innate and Sanofi to have focus on NKp46. The other companies have focused on CD16, NKG2D, which are less consistently expressed, particularly in solid tumors. So we believe that combining cells with NK engagers will provide optionality to clinicians, and particularly in those cancer patients that have insufficient number of functional NK cells. We have demonstrated that in preclinical studies and look forward to confirming that into the clinical studies. Again, this is our infrastructure. You can see our team in Natick, Massachusetts. We have capabilities in cell process development, in gene editing, in vitro and in vivo pharmacology, and then in Puerto Rico, not only for clinical, but in the future for commercial scale manufacturing. The management team is seasoned yet, very entrepreneurial. We're very proud to have Dr. Stanley Frankel as our Chief Medical Officer. To his credit, he has the approval of 2 CAR-Ts for Celgene, Bristol Myers Squibb. Immediately prior to coming to Cytovia, Dr. Whaley has built our R&D organization. And if you look on our board, we have people like Dr. Michael Friedman, who's a former acting commissioner of FDA, was the CTFO, was on the board of Celgene for 10 years. So in summary, when you look at Cytovia, Cytovia has first-in-class GPC3 targeting program for HCC with in vivo proof of concept and initial INDs first in 2022 and additional ones in 23. We're the first company to combine our own NK cell engager antibodies and iPSC-derived NK cells. We've integrated R&D and cGMP cell manufacturing, led by an entrepreneurial management team, an expanding network of academic and corporate partnership. We're the first NK company, at least from the West, and probably the first iPSC company to establish a Chinese-focused JV. And we have a broad pipeline expanding to multiple myeloma, glioblastoma, and potentially additional indications and targets through partnership with other biotechs and pharma companies. Thank you very much.